Good afternoon. I'm Paolo Cravedi. I'm an associate professor of nephrology at Icahn School of Medicine at Mount Sinai in New York. And today we are going to talk about transcriptomic signature, specially resolved analysis of the nephron structure in chronic kidney disease. So, kidney disease is a major healthcare problem. Over 800 million people worldwide have some sort of kidney problem, and kidney disease is the 11th leading cause of mortality worldwide. And importantly, up to 7 million of people die because of lack of access to renal replacement therapy, such as dialysis and transplantation. So the functional unit of the kidney is called the nephron. So there are about 1 million nephrons in adult kidneys, and they are composed of one portion that is called glomerulus and another one that is called the tubule. In the glomerulus, what happens is the major filtration of the blood and the formation of the initial urine. And then through the pass through the tubules, the urine gets concentrated, and the elements that are important for our organism get reabsorbed, and some other substances that have to be eliminated get released by this fraction of the nephron. And here is a picture at electron microscopy of one glomerulus. And it's important to keep in mind that nephrons cannot be regenerated in adult life. So we are born with a determined number of nephrons, and these nephrons have to be preserved and kept functioning for all the extent of our life. Importantly, if we focus a little bit more on the glomerulus, how it works, we notice that the glomerular filtration barrier, that is what allows the blood to be filtered, the urine to be filtered from the blood into the urinary space, is formed by three different structures. So one is the podocytes, which is a unique kind of cell that is in the outer space of the glomerular filtration barrier. And these cells are postmitotic cells, and so they do not proliferate. Then on the other sides, on the blood side of the glomerular filtration barrier, we have endothelial cells that are also unique because they present particular fenestration that are responsible for the first filtration activity of the glomerular barrier. And between these two cells, we have the basement membrane here that is a very thin layer of matrix that is composed by different chains of collagen that only the podocytes can produce. So we, as we said, we're born with about 1 million nephrons, and over time, patients that develop chronic kidney disease have a progression reduction of these nephrons, up to a certain extent that requires the replacement of the renal therapy either by dialysis or by transplant. And the overall idea of renal disease progression is that progressive loss of functional nephrons leads to the hypertension of the remaining one. And this initially represents a compensatory mechanism, but that over time, this leads to the loss of protein in the urine and progression of the renal disease. So it's important to highlight that the homeostatic balance between the glomerular cells and the glomerular basement membrane, or GBM, which is vital for the maintenance of the glomerular ultrastructural integrity and normal filtration function, is altered during chronic kidney disease, or CKD. And the treatment of CKD has been historically focused on slowing its progression by systemic approaches that are causing many side effects and are not specifically targeting the glomerulus, that, as we just saw, is the main functional unit of the kidney. So to develop new therapeutic approaches, it's important to get a better understanding of the glomerular biology and to have a better understanding of the molecular mechanism that determine progressive glomerular disease. It's also important to keep in mind that not all glomeruli and tubules are affected in the same way at the same time, even in the same individual, disease or tissue or gender. So there's really a variability across each nephron. So studying them all together, like here's a biopsy of a kidney that is highlighting the different structure, the different glomeruli and different tubules, does not allow to have enough granularity to understand the variability across the different glomeruli. And so by doing this bulk analysis that has been done until recently, we lose the information that is crucial to get the molecular mechanism important for the renal disease progression. So there are different techniques to get more insights about the single cells or single structure of the glomeruli. Here today we are talking about what is called the nanostring technology that allows to have transcriptional information of the different structure in kidney biopsy. And the way it works is by staining the biopsy, the renal biopsy, with antibodies that have oligo that are conjugated to these antibodies. Then the biopsy is analyzed, and we can define the regions of interest where we want to understand the gene, the transcriptional phenotype. And then once these regions of interest are defined, the biopsy undergoes UV cleavage of the oligos from the antibodies, and then these oligos get amplified, and so it will give us the information about the transcriptional profile of each single area that we were interested in. So we were interested in using this technology to better understand the renal disease pathophysiology, and so we started analyzing a few samples of patients with normal disease or with a genetic renal disease, with immune-mediated diseases, and with a primary renal disease that is not genetically driven. So the first disease we focused on is Alport disease. So this condition is characterized by abnormalities in the glomerular basement membrane and manifests clinically by the presence of proteinuria or protein loss in the urine, and the presence of glomerular scarring and hearing loss and ocular manifestation, because the same collagens that are present in the GBN that are abnormal in this disease are present also in the ear and in the eye. And here are the genetic abnormalities that drive this collagen disease, and can be, in most of the cases, X-linked, so associated with sex, especially in the male sex. But there are also forms that are autosomal and could be recessive or autosomal dominant. The second disease we focused on is membranous nephropathy. As you can see in this representative slide, membranous nephropathy is associated with a thickening of the membrane of the glomerular membrane in the kidney, and this is due to the presence of antibody against podocyte proteins. In most of the cases, against one podocyte antigen that is called PLA2R, or phospholipase 2 receptor, and the formation of the immune complexes in situ on the podocytes leads to podocyte injury and then proteinuria or protein loss and progressive renal failure. The third disease we focused on is called focal segmental glomerulosclerosis. So the name means that only some glomeruli, so it's focal, and some sections of the glomeruli, so it's segmental, are interested by the sclerosis. So focal segmental glomerulosclerosis. And so we know that the histological characteristic of this disease, as we just mentioned, but the pathophysiology, why this disease is happening, is still unclear. And similar to the other diseases, this condition is also characterized by proteinuria and progressive renal failure. So we decided to use these three diseases because they are really characteristic and of most of the glomerular diseases that can happen in the kidney. And here are the biopsies that we characterized. So we took 2 biopsies of patients with Alport disease, 2 from patients with focal segmental glomerulosclerosis, 1 patient with membranous nephropathy, and 1 healthy kidney that was obtained from a nephrectomy. So the renal function that is measured by serum creatinine was a little bit variable across the different conditions. But as you can see, almost in all these conditions, there were some degree up to a severe level of proteinuria. So we took the biopsies of these patients, and then we defined the regions of interest, or ROIs, that in our case were the glomeruli. So we picked the glomeruli in each of these biopsies. And then we took the tubular areas in each of these biopsies, with the ultimate goal to define the transcriptional profile in each of these single areas of each single biopsy of each single patient. And so we processed this data. Here are the details, how we did this processing. And the first thing we did was to make sure that the quality of our data was good. And we found that the sequencing depth was consistently high, yielding high confidence counts across all the samples. So the procedure worked just fine. And we also verified that there was a good dynamic range across all the samples in terms of gene expression. So here you can see the different diseases stratified by glomeruli and tubular areas, and each of these little dots represent one gene that is more or less expressed. And as you can see, there are genes that are blue and others that are white. So it means that there is a wide range of expression. If this range was more narrow, this would have limited our capability to capture differences across glomeruli and across patients. So this was the first time we used this technology, so we wanted to make sure that the data made some sense. So the first thing that we did was to compare the expression of genes in the glomeruli versus the tubules. And we were happy in noticing that the glomeruli were expressing uniquely podocyte genes that were not expressed in the tubules. And they were also, again, the glomeruli expressing genes that were characteristic of glomerular endothelial cells and mesenchymal cells that can be found only in the glomeruli, not in the tubules. We next put together all the tubules and glomeruli from the different patients. And again, we were reassured in seeing that in this principal component analysis that represent in a two-dimensional way all the different dimension represented by the expression of each single gene, that the tubules cluster different from the glomeruli. So indicating that the gene expression profile of the tubules and the glomeruli was different as predicted, as these are two very different regions with two different structure and different functional capacities. Importantly, we also noticed that in gray you can see the normal tubules and normal glomeruli from this normal biopsy, and all the other ones, the other squares and dots and triangles that are colored are from patients with diseased kidneys. And the gray and the colored dots really well segregate one from the other, so indicating that there is a difference as predicted between diseased and normal kidneys. We next went more granularly and we tested each single disease. So we started from Alport disease, the genetic disease, and it was compared against the normal glomeruli. And as you can see here in the square, the tubules segregate differently from the tubules of Alport patients, and the dots again were well separated from the dots of the Alport patients. Interestingly, we also noticed that in light green and in red, we represented the two biopsies. One was from a male, one from a female that has slightly different genetic abnormalities. And so we were able to appreciate the difference between the two biopsies. And within each biopsy, each of these dots represent a different glomerulus. So you can see that even though all the glomeruli from the same biopsy cluster in the same area, there are differences across different glomeruli within the same biopsy, which made this kind of analysis very, very interesting. And we next we did these volcano plots that allow us to define the differences in the expression of genes. So on the x-axis, we have the fold change. So the more they are on the left or on the right side, the higher is the difference in the expression between the normal versus the Alport glomeruli. And on the y-axis, we have the statistical significance. So the ones that we have in the top left and top right are the genes that are more more differentially expressed between normal and Alport syndrome. And we were able to identify some genes that we knew being differentially expressed. And importantly, we found differences also between the two patients. When we compared the two patients, the male and the females, more in detail, we were able to identify to pinpoint the single genes that differed between the two patients. We did the same thing for focal segmental glomerulosclerosis. Again, here we have the glomeruli of the FSGS patients, and in gray, the patients, the normal glomeruli that segregate differently. And here are the volcano plots all together on the left, in the middle for the male patient, and on the right for the female patient. And if you look at the volcano plot on the left, putting together both biopsies, you can see that the most highly significantly upregulated gene in the FSGS patient was the C3, and we were pleased in seeing C3 because it's one of the complement components, and we have been interested in studying complement in this condition, and indeed it's not uncommon to see complement deposit in FSGS, nor in the plasma of patients with FSGS. And in patients that have low C3, so that have a consumption of complement, there is a lower survival of the kidney in patients with FSGS. So overall suggesting that complement is important in the pathophysiology of this disease, similar to what this spatial transcriptional analysis was indicating. We arrived to this conclusion through a different path, but is really intriguing that the same path is independently by this spatial transcriptomic analysis. And to formally prove that complement is implicated in the pathophysiology of the disease, we and others have done some mouse experimental data in animals that don't have C3. And as you can see in green, the mice that don't have C3 have less albuminuria or proteinuria or disease severity than wild-type controls upon exposure to adriamycin, that is one of the strategies to induce FSGS in mice. We next move to our immune-mediated disease, membranous nephropathy. And here we also found a pathway that we were not really aware of, that was upregulated, the Il4 pathway. Here you see in the Volcano plot an upregulation of one of the genes of this pathway. And in the expression pathway, again, IL-4 ranked amongst the top pathway. And when we looked into the literature, we found that indeed Th2 cytokines, so cytokines by T cells that that produce IL-4, were characteristic of patients with membranous nephropathy. So again, these data with the spatial transcriptomic were partially confirming the previous literature, but also pointing at new genes that in the future we will be interested in studying more. So the next step for this kind of study will be to assess the differences, not across biopsies, but across the various glomeruli and tubules, which was the main initial reason why we used this technology to start with. And as a step further, we used a computational so the question is, are there differences across cells within a single glomerulus? And to answer this question, we use a computational strategy to make a deconvolution of the cells that are present in each glomeruli and in each tubule that we selected in our region of interest. So using this computational algorithm, we were able initially to define differences in the cell composition between glomeruli and tubules, which was expected, but was kind of our control that this algorithm was working just fine. And here, just by looking at the composition of the different cells in the glomeruli and tubules, we were able to identify, for example, in the glomeruli, some cell subset that were not really expected to be part of the glomeruli, and similarly so in the tubules. And if we just look at the composition of the different cell subset in the glomeruli and in the tubules, we were able to identify already some differences across the different biopsies. And this was true for the glomeruli as well as for the tubules. So the next step here will be to assess the gene expression profile, not just in each glomeruli, but in each cell subset within a single glomerulus, which is expected to give us important information about the variability across the different glomeruli, understand whether glomeruli that have similar histological feature have a similar transcriptional profile, or vice versa, if glomeruli that look different have similar transcriptional pathways that are activated. So that will be, we believe, very important to better understand the molecular mechanisms that are responsible for the renal disease progression. And finally, together with the transcriptional profile, we also tested the protein expression profile within the kidney. So the same slides, actually serial slides, were also stained for different markers, protein markers. And the purpose of this staining was to, on one hand, validate the transcriptional profile, so that if we saw upregulated gene, we could confirm that also the protein were upregulated. But the other idea was also to better define the single structure in the kidney based on the expression marker that could allow us to identify regions, smaller regions of interest to test their profile within these regions. And these are the preliminary data that we obtained so far with this technique, where there are many proteins of these panels that are not really over or under expressed, but there are two subsets of proteins that are highly expressed or express or lowly expressed, that will be informative to better understand and integrate the transcriptional profile data. So overall, to conclude and summarize our data, we observed differences between Alport, FSGS, and membranous glomeruli, as well as between glomeruli and tubules within each disease. We found that not all glomeruli or tubules are affected in the same way, even within the same kidney, and the spatial profile can clearly help us understanding such differences. It also seems that males and females are often affected with CKD differently, and this was clearly observed in our Alport and FSGS samples. This could be attributed by the variability across samples, but could actually be something that is really sex dependent. So understanding their differences at transcriptional level and also at the protein level could lead to more specific and personalized therapies. So in conclusion, the spatial profiling of gene expression we think is a powerful tool that will allow us to answer some very important questions about the mechanism of disease progression in glomerular and CKD, such as Alport, FSGS, and membranous nephropathy. Thank you for your attention.
Okay, awesome job. Well, we're all set pretty much from here. I will do some quick editing on the front and back ends, and then we will get your presentation ready for the event.
Okay, excellent.
Awesome. So any questions for me before we wrap up?
I think I'm good. So on my side, what I have to do is just to give you the numbers of the slide that I would like not to be included in the PDF, correct?
Yeah, you can do that, or you can edit your PDF or your slide deck and remove the slides that you don't want. You can send me over that slide deck and I can use that.
Okay, that's probably easier. Okay, let's do so.
Perfect.
That's it. And you're gonna send me like the link to my presentation, and that's it.
Yeah, I can send you a preview link once we're done with some editing.
Okay, excellent.
Awesome. Alrighty, well, we will talk to you soon. Have a good rest of your day.
Bye. Bye-bye.
Bye-bye.